have we're hoping to have a members only show and tell and virtual holiday social in the first week of December, but more on that as well as an upcoming general election. So Laura.
Hello, everyone. Welcome. I was going to say for many of us in the map world. Hello. Can everyone hear me?
Yep, we got you.
Okay, great. I was going to say for many of us in the map world, John Hessler is a legend and he needs no introduction. He's a mathematician, a GIS scientist, and a professor, and also the author of more than 100 articles on map-related subjects. He has written two of my favorite map books, the first one called Map, Exploring the World, a Phaedon book, and Seeing the World Anew, the Radical Vision of Martin Wall-Seemüller's 1507 and 1516 world maps. John is the founder of Lambda Lab, whose theoretical work currently centers around using innovative mapping methods like bioacoustic computer vision and on the development of new ideas in evolutionary and algorithmic game theory for complex policy analysis. The lab is a member of the BAT1K Genome Project, whose current research focuses on using bioacoustic recordings along with wavelet analysis to study the spatial distribution of bats, the hosts of SARS-CoV-19. We are so honored tonight that John will join us to discuss the most sensitive and important topic of our time. It came from a blue sky, mapping bats and spillover of COVID-19. Welcome, John.
Welcome.
Thank you very much. Thank you everyone for inviting me. It's been a long time since I've talked to the New York Map Society. I think the last time was at the Ethical Culture Society. It was during a giant snowstorm, probably 15 years ago, maybe even more. It's great to be back. Tonight's going to be a very different talk mostly than I think most of you are probably used to. We're going to be really getting down into the weeds on some very technical mapping and some techniques that are taking place today. Some of the research that I am doing, some of the research that my lab is doing, and some of the research that others are doing, which we'll talk about as we go through. We're going to concentrate really on what, as Laura said, a central issue in our world today, which is the zoonotic spillover of SARS-CoV-2. And really what we're going to zero in on tonight is really what mapping bat biogeography, what the spatial variation in bat genomes can tell us about this spillover. And we're really going to, as I said, by the time we get to the end of the talk tonight, I think you'll have a real clear idea of where this virus came from, and we'll basically kind of tear apart some myths that are sitting out in the press and some of the conspiracy theories, and kind of replace them with some facts. So if you don't know too much about zoonotic viruses, in the last few decades, there's been a lot of them. Nipah, Hendra, Ebola, Marburg, HIV-1, HIV-2, SARS-CoV-1, and of course, the more recent COVID-2 are really zoonotic viruses. They're really, for the most part, RNA viruses, which they have very small genomes, really ranging only into the tens of thousands of nucleotides, and we're going to talk a lot about that. By the time we get done with this, you're going to kind of be an expert on bat genomes and the important parts of the genome that we are looking for when we're talking about sampling bats for SARS-CoV-2. But really, this is a phenomenon that in the last few decades has become really important, as have this emerging RNA viruses, which you will also come to know quite intimately in the next half an hour or so. So if you've never been in an actual bat cave, so this is what it looks like. One of the questions that we're going to kind of address tonight is, why is it that bats seem to harbor so many of these viruses and don't get sick themselves? This has been a complicated question in biogeography and ecology for a very, very long time, not just in recent years. But in the last 2 years, of course, this has become much more important with the COVID-19 pandemic and people looking a lot more closely about what it is about the immune system of bats that allows them to harbor so many virus and host so many virus and not get sick themselves. And that's really still quite a mystery. This super evolution, this super immunity to viruses seems to have come about from about the time that they developed flight. It's a long evolutionary path. But really, a lot of the ways that the virus keeps itself together is basically passed on from bat to bat. There are not a huge percentage of these bats that you see now that are going to be infected with a particular virus, but based on their lifestyle, based on how they live, you're going to see that only a small number need to harbor the virus to keep it going in a population. And so that's really what we're going to center on tonight. We're going to really center on how it is that bats harbor these viruses. How do we know where these bats are? How do we map them? How do we trace the biogeography of bats? Because one of the things that's very complicated is that we don't know a lot about them. We really don't know a lot about the ecology of bats. We certainly don't know a lot much about the viruses that they harbor. Very few have been sampled and not transfers from bats. And as I said, we'll get deeply into that. So when we're talking about bats, there's a couple of different suborders of bats, and one are called microbats and one are called megabats. And what we see here is in the yellow are the megabats. You can see their distribution is mostly Africa and into Asia, and then up into the southern Europe and a little bit into England. But you see in our area, in our hemisphere, there are no megabats for the most part. We mostly have microbats, which for strange reasons that are phylogenetic, which we're not going to go into, the family Rhinophidae and a few other families are included in Old World megabats, even though they're small bats, they're not large bats. And really the family that we're going to be looking at, and the genus we're going to be looking at specifically tonight, are Rhinolophus bats, which are the ones that are believed to harbor, and by the end of this, you will see proof of harbor SARS-CoV-2. And so we're really talking about an old world bat. We're talking about, as you can see in the map, the distribution lines across there. These are some of the migration paths and some of the things shown for New World and Old World micro and megabats. But for some reason, just I said, phylogenetically, which we won't go into, the genus of bats that we're going to be talking about is in the megabat family. Most megabats are fruit-eating bats, but we really are going to be talking about insect-eating bats in the Rhinolophus genus. Again, this is the sort of major distribution lines of megabats. Some of them eat fish. Most megabats have great eyesight, as opposed to the Rhinolophus that we're going to be talking about, which basically navigate in the way you would typically think of a bat, through echolocation. And you can see that the distribution of these, as people have mentioned and people have talked about, the origins of SARS-CoV-2 being in China or Indonesia or out somewhere in that region, is really the main place that we find these kind of bats. And so that at least is one idea we know, but we really don't know the specific biogeography of these animals. And this is really an important point. So obviously SARS-CoV-1 and SARS-CoV-2 are very similar, and this kind of led researchers to begin searching among bats for possible links. The Rhinolophus genus of bats, they're called horseshoe bats in common parlance. They're divided into 6 subgenera, and there are many very small groups. They have a long common ancestor, 30 to 40 million years ago. But it really is unclear where the geographic roots of this family are. And really the attempts to determine its biogeography have been really difficult and really hampered by the mostly unknown ecology of this genus. And so one of the things that my lab has been looking into, and we've been looking into, is really trying to determine some way to map what the distribution of Rhinolophus bats are, and really what the distribution of the specific species of bats that we've been able to sample the genomes that are related to SARS-CoV-2. And you'll see that there's a wide gap between some of the bat SARS-CoV-2-like viruses and the human version of SARS-CoV-2. And we'll talk about that evolutionary distance. But one of the most important things is really to somehow determine where these bats live, what their ecology is, and begin the process of more field sampling so we can really understand what their immune system is and what viruses they harbor. Obviously, these types of bats are coming into contact with people more and more with global warming, with the change in their habitat, the loss of their habitat, and for some other reasons. One thing that has been talked about in the press is, of course, the Wuhan market. So the first question we'll ask ourselves is how did the zoonotic transfer of SARS-CoV-2 happen? And a lot of people have talked about the Wuhan market as the place that this may have taken place. Most scientists at this point are basically questioning that in a very, very deep way. And they're doing that from a couple of studies. The most important one, the one that I have in front of you now, which is called Animal Sales from the Wuhan Wet Markets immediately prior to the COVID-19 pandemic. The scientists who put this together basically had access to all of the shops across Wuhan's animal markets. So 17 different shops. They reported a total sales of 36,295 individuals belonging to 38 terrestrial wild animal species, and averaging about 1,170 individuals per month being sold. This is an important study because it really came up to the study was accomplished right up until the point where we see the beginnings of the COVID pandemic. This report that you're looking at here was just published a few months ago. It took some time for them to get their data together. But this is really the most profound study of all of the animal sales in the Wuhan wet markets. It's really important that this was just happening prior to the COVID pandemic because it shows kind of trends in what was being sold. What's important to note here is that there was not a single case of a bat being sold in the Wuhan wet market. And if bats are in fact the source of SARS-CoV-2, they weren't present in the wet market before the COVID-19 pandemic began. There has been talk about an intermediate host, something like a pangolin has been talked about. There is not a single pangolin that was sold in the Wuhan wet markets immediately prior to the COVID-19 pandemic. So there's a lot of work that needs to be done on what role the Wuhan market may have had. People are, of course, pointing out in a more detailed way at this point in time that this may have been more of a super spreader event than where the zoonotic transfer actually took place. So from that perspective, there's a lot of different species that were being sold, many rats, many birds, lots and lots of mammals, none of them in the greatest conditions, but the animals that we were really pointing towards, at least bats, were not sold in the Wuhan markets. And so that is something that I just wanted to get out there. It's an important point. But bats still are the place that we are looking to discover the actual source of SARS-CoV-2. Now, when we talk about this Rhinolophus tree, the Rhinolophus bats, there are lots of species in the Rhinolophus genus. They're across China, across Central Asia, across Laos, into down along the southern islands, into India. And even two species that actually found in Great Britain and Europe. Now, the species that have been implicated in the SARS-CoV family, so either SARS-CoV-2 or SARS itself, SARS-1, come from really across the phylogenetic tree. So these bats are from all over the sort of genetic and genome of this set of species. So really across this genus, we see samples of SARS-CoV-2 within these bats. Now, one of the things that's very important is when one is actually looking for signs of a virus, antibodies don't necessarily give you much, aren't much help, but live virus is. SARS-CoV-2 live virus has been sampled from several bat species. You can see two here, the Aphaenus and Malleanus. Live virus has been sampled, and we'll get into what that means. Now, that's not live human virus, that's a SARS-CoV-2 virus. That is a bat that's very close to our genome that's sampled from a bat. And so basically, you can see from across the phylogenetic tree, there's basically species from all over the genus that have the SARS present in them. Most of the other ones haven't been sampled, so we don't know. And so really, there's a real mystery about how extensive the SARS type species in this particular genus. Now, what's important about this from a mapping perspective or from a geographic perspective is really what this shows us is that this we can't be looking for a single species necessarily. We're kind of looking across the phylogenetic tree here. And all of these species don't necessarily exist all in the same place at the same time. So if we can narrow down which species actually are the ones that are of interest, we can kind of begin to get an idea of what we're looking for. But the question is, how do we look for them? It's very difficult, and we're not going to send someone in a secure suit into every cave and every nook and cranny all over China and all over Vietnam and Laos and India to see where these bats live. So the real question is, what is the distribution of these bats? I mean, we see animal distribution maps all the time, but how do we determine where they are and what their lifestyles are? And to do that, we kind of have to look to different means, different ways of doing things. We obviously can't go into every cave and band every one of these animals. Rhinolophus bats also move around quite a bit. Some of them fly 40 or 50 miles in a night in search of their food. And so they roost together with other species in caves all over the place. But so how do we determine where they live, what the extent of their reign is, and produce a map that says, here's where Rhinolophus aphaenus lives, here is where Rhinolophus malleanus lives. And so that's sort of the problem. That's sort of the issue. These things are in the area of interest, where people are saying Hubei province. These particular species of bats that we found, that have been found to have SARS-like viruses, are well represented in the area where people believe the virus originated. Now, again, we have to go back to the fact that the Wuhan market didn't seem to have bats in it, but people in China and some of the rural areas here are in contact with bats quite a bit. There are the guano people who go into the caves to get use the bat species for fertilizer. There are religious ceremonies. There are Buddhist caves which contain millions of bats. There are tourists who go into bats and into caves to see the bats. There's lots of contact with bats, which doesn't necessarily have to be within a market or within a food or in a food situation. It's dramatic to think that, but it probably isn't the place where the zoonotic transfer took place. It probably took place in much more humble circumstances and less dramatic circumstances than we might initially be led to believe. So how do we begin to figure out where these bats are? So really what we've started was a few pilot projects using remote and real-time bioacoustic sampling. And basically what this allows us to do is put bioacoustic recorders in the mouths of caves and all over the areas where bats are supposedly known to roosts. And basically over a certain amount of time, record the sounds, record what is actually going on, both in real time and remotely. And we can take those ambient recordings, and basically this is what one looks like. and that's of course me in a cave putting one in. So this is actually an ambient recording. Now this has been, the frequencies here have been changed so you can hear them, because obviously bat echolocation calls are, can't be heard by human beings. But you can see, as you can hear the sounds, you hear lots of background noise, lots of background high frequency noise. But those little chirps that you see, that you hear, are actually the echolocation calls of a bat. Now, each of the species of bats has a slightly different echolocation call. And when we look at our Rhinolophus friends, this is just a plot of a time series plot of the amplitudes of 4 different species. These are 4 species that are thought to harbor COVID-19 or SARS-CoV-2. I'm going to use those 2 interchangeably, unfortunately. Actually, SARS-CoV-2 is the virus that causes COVID-19, but I seem to use them interchangeably, as does everyone else. So you'll hear me use them both. But these are the 4 species that that people think might have some implication in the zoonotic transfer, at least did. And this is the amplitudes of their echolocation calls. So you can see all 4 have very different patterns. Now, this is just amplitudes. And really what we want is we want to understand the frequency of these echolocation calls. So when we turn these into frequencies, we can see that Rhinolophus bats use what's called a constant frequency echolocation call with a little bit of a moderated end. Most bats will use a frequency modulated call. But this particular genus of horseshoe bat uses a constant frequency echolocation call. And so you can see on the right-hand side there, the left-hand side is the conversion of the signals that I showed you earlier into actually the frequency spectrum. And then on the right-hand side, you see the various forms and the various amplitudes and frequencies of each of the species of bat. So each of the species has its own location, its own kind of form. There's a real fingerprint almost to the bat calls, but they overlap a lot. They're very close to one another. So the question is, even if we do these field recordings, and let's say we take 1,000 hours of field recordings over a wide swatch of territory where people think a Rhinolophus affinis group lives, we still have to somehow be able to take that information, analyze it, and figure out what those sounds actually are. There's a lot of noise in those sounds, as you saw. There's a lot of beeps and a lot of, even running water has a high frequency component to it that interferes with these sounds. So how do we actually figure out how to fingerprint the sounds that each of these species make? And so this is where we turn to a really complex way of looking at these sounds. So here is just gonna. This is the call of a Rhinolophus bat, just so you can hear. It's got this two little chirps in it. But how do we, and this is the actual spectrum of this bat flying. So obviously you can see it's echolocating, it's pushing on the bottom. On the top here is the amplitude. So this is the amplitude of its echolocation, and this is the frequency spectrum. So it's chirping, 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 chirping. So it's echolocating. These will get faster and thinner if it's approaching an object, or if it's hunting, or if it's looking for an insect. So how do we take this information? Now, we know this signal is from a Rhinolophus bat. What do we do to separate this out from other bat signals and other species that will allow us to actually map on a map that you're familiar with, the actual locations of where these bats live? And one of the ways we can do that is through wavelet analysis. Now, what wavelet analysis does, it allows us to break the spectrum, the amplitude and frequency spectrum of the Rhinolophus bat signals into smaller and smaller packages. So this is basically taking something and looking for fingerprints at various scales. So we're familiar with regular normal mapping where, you know, if you're looking for a street someplace, you're not going to pull out your world map. You're going to pull out a map at a very high scale so you can look at the details. What this allows us to do, what wavelet analysis allows us to do, is it allows us to look at very, very fine details within a signal and see if we can find particular fingerprints. So up at the top here, you see the Rhinolophus signal, and these are all of those echolocation calls squeezed together. And then we can break it up into what are called wavelet coefficients. And we can look for particular fingerprints of each particular species at very, 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 very high scales. And once we do that, we can teach a computer to take signals, many, many thousands, many, many hundreds and thousands of hours of these signals. It has learned what to look for. So basically, we've trained a neural network, an artificial neural network, to recognize these signals at the various scales. And it can begin to take those signals and then begin to basically, in the frequency spectrums or in the recordings that you saw, this is again I'm just going to stop this bat from squeaking here. Sorry. And so basically what the machine learning program does is it takes those thousands of hours, it then finds the actual signals in the spectrum that are BAT related because it's learned exactly what to look for. And then it begins to ID those based on the very small scale spectrums, the very small scale things that we can find within the frequency spectrum and the amplitude spectrums of the BAT themselves. So this process of putting out very inexpensive field recorders, leaving them out in the field for thousands of hours in areas suspected, basically allows us to begin to at least begin to explore the geographic space that these bats inhabit, and allows us to begin to map some of this material, some of where they come from. Now, this is important for a number of reasons. So what we've talked about so far, obviously, is this idea of using these bioacoustic signals to actually map the biogeography and the distribution of these bats. What early experiments have shown us is that many of these bats roost together. These species will roost together in the same caves. They fly fairly long distances. And we still don't have an accurate map, and probably won't for many years, of where these bats are. But what we do have is we do now have a method, at least, to go to a place where if there's a large cave, we can actually put these acoustic recordings out there and begin to determine what bats are there, where they're going, and put recorders around in order to begin to determine what the spatial distribution of these animals are. Now, why is this important? And this is really the central question, at least for me. Maps to me are a solution to a problem. They're not just a decorative thing to hang on the wall. And once the problem is finished, they're just a piece of data like anything else. But why is this piece of data important? Why go through this trouble of spending all of the time that it took us to come up with a wavelet analysis in order to be able to dissect some of these signals, and then to train an artificial neural network to be able to recognize them? Why is this important? Well, it's important for obvious reasons. We really want to know a couple of things, and we're going to take a little side trip here into SARS-CoV-2 and into the BAT part of SARS-CoV-2, and why we need to know which bats are where and why that is important. And so obviously, one of the things that you've probably heard in the news is this spike protein, okay? The spike of the SARS COVID virus, which really is about between 23,000 and about 25,000 in its nucleotides. So the entire SARS COVID virus is only about 29,000 base pairs. So it's very small, it's not very big. The spike protein is the most important thing. It is really where the virus itself binds to the ACE2 human receptor binding domain. It's extremely important, and it's a very variable part of the genome. So for a virus to make a zoonotic transfer, to make a jump, that particular spike protein is going to have to bind to a human ACE2 receptor. And the question is, which bat species, if we locate the genome, if we figure out, if we capture a bat, we sequence the virus genome that we find within the bat, how close are the bat SARS related viruses to the human the human SARS viruses? And are they close enough to be able to bind to an ACE2 receptor? Or is there an intermediate host that the bat has to transfer it to, and then that host transfers it to the human? And that really is the open question. And the reason that we are spending so much time mapping is because once we do this genomic work, which as you'll see in just a bit, is really zeroed in on some specific places. Once we do that genomic work, we really want to know where those bats are, and we really want to be able to go out and sample those bats to see how widespread this particular virus is, and perhaps other viruses which are possible candidates for zoonotic transfer. Zoonotic transfer is an extremely rare thing, but obviously based on what we see now, what we've seen with things like HIV, Nipah virus, Marburg virus, Ebola viruses, they're very serious when they do happen. And so we want to really know as much about first the genome of the animal, but then where is the animal and what other animals does the animal interact with? So that's why the mapping project is central to this. Now, when we talk about the closely related SARS viruses, there's a sample called RA-TG13. So RA is for Rhinolophus affinis. It was a sample of a coronavirus that was taken actually in China in 2013. No one really cared about it until now because it was just not all that interesting. There was no SARS-CoV-2 at the time in 2013 to compare it to. So this particular bat sample, obviously people are now going back and started looking at the samples that were taken and the genomes that were done earlier, but this is 96% similar to the overall SARS-CoV-2 human version. Okay, so we have a 96% match in this particular virus for RA-TG13. Okay, SARS-CoV-2, our SARS-CoV-2, the human version is really optimized for binding to the ACE2, and the spike protein is really where the binding takes place. It doesn't appear as if 96% to your ears, 96% seems like, wow, that's really close. But that's a lot further away in genome sequencing than we are from a chimpanzee. So it's quite a distance away. No one believes that there's a possibility of a zoonotic transfer directly between, say, something like a Rhinolophus affinis and a human. But this is a really important sample that got a lot of press very early on in the pandemic, and it's still an important sample of the virus that's found in a Rhinolophus bat. So this is from China. Again, Rhinolophus affinis, an important species. You heard the sound of the Rhinolophus affinis just a little while ago when I was playing it. But we don't really think at this point that that's the real place to look. Now, there's another species which we talked about earlier, Rhinolophus malleolus. And again, there's a sample of RMYN02. And this actually came from China in 2019 and 2020. This is one of the closest relatives to the human SARS-CoV virus. Now, when we talk about phylogenetic trees, so this is a phylogenetic tree. So basically what this is this is the relationship of the genome of all of these particular animals that have been sampled. So up here at the top is SARS-CoV-1. And you can see here that right at the top here, the closest relatives to the human SARS are these particular species of bats. We have our friend Rhinolophus affinis here. When we go down to this part of the tree, obviously SARS-CoV-2 is very different than SARS-CoV-1, so it's a distance apart. But these trees also have a geographic component. These are in fact geographic maps in very many ways. And what we can see here, and I'm going to show something at the end a little bit different that shows how to map the dynamics of this sort of thing. But we see here these samples from 2019, 3 2020, and 4 2020. So this is from Wuhan. These are from the samples of human SARS-CoV-2 taken from Wuhan. And then we see right next to this, the closest relative to that in the animal kingdom is our friend here, RMYN002. So very, very close. We see the next closest one, our friend RATG13, Rhinolophus affinis. Now, these are two different viruses. Their genomes are slightly different. Obviously, they're on different parts of the tree, but they're very, very close to the Wuhan samples. Of course, we don't know what we don't know. So these are samples that were taken. There are not that many BAT samples of SARS-CoV like viruses available for genome sequencing. They're getting many, many more now. But we can see these are the two BATs that seem to be the most important ones, at least at this point in time. And so this is going to change, and this has changed just in recent weeks. There's been new research that has changed this information, which you're going to see in a little bit. But these are the two kind of BATs that we really initially wanted to focus our analysis on. And we were able to actually, using wavelet analysis, to be able to split the calls, even though they look very, very similar in their amplitudes and in their frequency spectrum. We were able to find specific fingerprints between the two, which could be recognized by a machine learning program. So we were able to actually accomplish that task and begin the sort of process of beginning to map the distributions and to use this kind of tool. Now, when we talk about the SARS-CoV virus, this is as we said, it's an RNA virus. It has a small genome. It's about 30,000 nucleotides. Is really more than 1,200 nucleotides, and that's quite a distance. So we have to look for something a little bit different. And a little bit better. So that's kind of the next step. Now, when we're talking about the genome of the BAT viruses, really what we're really looking for is the genome of this bit up here, which is the spike protein. It's the thing that makes the coronavirus look like the crown. These little things popping out, which are these spike proteins, these spike glycoproteins. Here we see an actual image of SARS CoV-2 within a cell, having bonded to the cell. So what happens is that this bonds to a human cell, and then that allows the RNA from within the viral capsid to enter into a human cell and begin its replication process. Now, the phylogenetic tree, we can actually just look at the spike protein part. And that's kind of important because the spike protein is the place that we really want to concentrate on, because that's the thing that is going to allow binding to take place. So if we just look at the spike protein, we can see that our friend RJTG13 is much closer than our MYNO2. So we really are still focused in on this Rhinolophus affinis species as the major candidate, but it's still only 96% overall of the overall genome, similar to the human version. Just going to skip this. So it's kind of improbable, and we'll look at this in just a second. We have this situation at this point where we have these two BAT viruses, which are very similar to SARS-CoV. They're close enough that people have been looking very closely at them. And really what people have really been looking at is this area, this binding area to AC2, which is called the receptor binding domain, okay? The SARS-CoV-2 is optimized. It's really good at binding to the human ACE2, but it doesn't really seem to, the BAT ones are a little bit different, which has of course led to this idea that it was manipulated in the laboratory. Molecular biologists are not God. They can't create viruses out of nothing. SARS-CoV-2 is a completely new virus. It seems to have come from 5 natural recombination events, which means pieces of the genome recombine naturally within the BATs in order to make this particular virus. And so it's really important that we kind of look beyond that conspiracy theory, because it just doesn't seem possible that this new virus, which is optimized for this receptor binding domain and is the product of 5 recombination events, and I'll tell you what that is in just a moment, could really have been engineered in a laboratory. And there's new data, which I'm going to show in just a moment, which really definitively proves that that is not a possibility. So basically, we have these two things. The RMYN02 shares 93% of the identity with SARS-CoV at the scale of the complete virus genome, but in fact, it doesn't show anything close to the sequence in the receptor binding domain, only 61.3%. So we can kind of throw out that as a candidate for really the zoonotic transfer of SARS. So in the genus Rhinolophus, they're obviously important reservoirs for coronaviruses, and they currently harbor the closest relatives to SARS-CoV-2. But the observation that it contains these insertions, these amino acids in the spike protein, really indicates that they're natural events. And I'm not going to go into the details about that. I'll send a link around to the hosts, and they can send you can go to it to kind of read a little bit more about that. But now, if we look at the geography of these two bats, these two Rhinolophus and Rhinomelanus. So one is found to be 96% similar, and the other is found to be 93% similar. Other bats have been sampled over the years. You can see on the list here, the 2020, 2019, 2018, 2013. So over the years, all the way going back to 2010, we have genome sequences. All of these particular species of bats harbor coronaviruses. You can see what the difference in the location. So RGTG13 came from Yunnan, and we think that Hubei is the place where the original outbreak happened. And so you can see the sort of distribution here. Pangolins are thrown in here just for comparison, that they're kind of distant in their COVID or coronavirus genomes from very far from the human, further than the bats, showing us something about the lack of possibility of pangolins being involved in the transfer here. Now, just a few days ago, there's been some more data put in, but just to kind of summarize where we've been here, the spike protein sequence appears essential. Okay, it determines the binding affinity of the receptor domain, and is therefore responsible for the host range. So what that means is, what we're really looking for and what we're really trying to map is animals that have a spike sequence that is very close to human sequence in the receptor binding domain, and that it's possible to bond to the ACE2 receptor. The closest bat strain to date was RATG13, but has a low sequence similarity to SARS-CoV-2. Only 11 of 17 human ACE2 contact amino acids are conserved with the human version, which means that this particular bat really doesn't really have a possibility of really binding with the human ACE2. So the SARS-CoV-2 poorly infects bats and bat cells tested so far. So the essential piece of information that we're missing, and the thing that would drive the mapping project, and up until this point drove the mapping project, was really finding bat viruses with a receptor binding domain motif close to SARS-CoV-2 and binding to the ACE2 with high affinity. And at this point, at the point where this was actually written, this was missing. And so the mapping project kind of went to just look for various species of Rhinolophus bats until 2 weeks ago. Several weeks ago, a paper was published called Coronavirus with SARS-CoV-2 like receptor binding domain, allowing ACE2 mediated entry into human cells isolated from bats of Indo-Chinese peninsula. So this is going to be a really important paper. It was actually done in preprint. It's being peer reviewed at Nature at the moment. The New York Times actually picked up a story just a few days ago on this particular paper. And what we've have basically shown, and we're going to get into this in a little bit of detail, is that there's a group of bats. You can see the sampling sites along the Mekong River here, site 1, site 2, site 3, and site 4, where they sampled bats from caves, Rhinolophus bats from caves. These were all Malleanus species. There's a Marshali species and a Pusillus species. So again, we're increasing the number of species of Rhinolophus bats that seem to harbor COVID viruses that are related to SARS. These particular viruses, it turns out, are very close to the human spike protein, and this basically shows the similarity structure. We can see that our 3 or 4 of our banals here, these are the ones that we're talking about, are much closer than our friends, the RAs and the RM. When we look at the various sections of the genome, and again, the most important point is, of course, this receptor binding domain, which is 11, okay? So if we look at our phylogenetic tree, we can see here in China, Wuhan, here's samples taken from people at the Wuhan seafood market. All of these are geographically related to the Wuhan area. These are, of course, human. And then we've got the bat viruses. These are the ones that were isolated from Laos right here. And then here's our friend RA. So the 4 closest are the ones that have just been isolated from Laos and the Malleanus. What's really important, however, is basically the receptor binding domain closest to that of SARS is now in 3 different bat species from Laos, Marshali, Malleanus, and Pusillus. Obviously, Malleanus has been implicated in a couple of different things now. The results basically support that SARS-CoV-2 could originally result from recombination sequences pre-existing in Rhinolophus bats living in limestone caves in Southeast Asia and South China, and provides the ideal conditions for interspecies interaction. So what that means is Rhinolophus bats of these species interact with one another. They forage in the same caves. They're constantly interacting one another. They're passing the virus back and forth. And there are these recombination events, basically where the virus is recombining in each of these animals to produce this virus, SARS-CoV-2, with a very close receptor binding domain to the human version. Okay, the ones that have been isolated share 16 to 17 of the human ACE2 contact amino acid residues. This does not appear to require an intermediate host. The researchers actually were able to bind a sample of the bat virus to human cells. So bat to human transfer is completely possible, a naturally evolving thing in the world. So the next step here obviously is to look for these 3 species of bats, and that is the next adventure in sort of mapping research using the techniques that we've developed. And there are several researchers who are taking these into the field with rather large scale recording devices in order to try and figure out whether these techniques will actually work, whether we can actually determine the differences and the actual distribution of these and map them on a regular map. So that's really what I have. I'm going to pop out of this for a second because I want to just show you a particular something else before we close, and then I'll take questions. So one of the ways, and this will be the last thing we're going to talk about. So what I'm showing here is I'm showing a basic breakdown of the current genome of the human viruses. So what we can see here is we can see as time goes on, this is March 2020, this is the phylogenetic tree of the various human viruses. Here's the 21J delta, which is responsible for all the cases in the world at this point. One of the things that we've been doing with the human version, again, this is different from the BAT version, is actually mapping the actual transmission through time. So if we, and I'm just going to filter this a bit. And so these genomes are very, as long as I can get this to play. Come on. Let's try it again. All right. So basically, using these genome sequences, we can begin to actually look at how and where these things develop. So I'm only mapping right now. What you're going to see on this map is just the rise of the delta. And obviously, as we're coming through, you can see we're going month by month. Sorry about that. We're going month by month here in the date range, and we're now in. So I'm just going to speed this up a little bit because it's not behaving well. So we can begin to actually, all right, come on. So we can see, we can begin to map the first cases of the Delta as it spreads around the world using these genome sequences. And so these are the kind of maps that we're also developing to basically look at how the various variants spread around the world. Like I said, this is not working and it's going to drive me crazy here. So I'm just trying to get a date range that works. Okay, let's try this again. So we can see that these are the, no, it's just not working for me. So in any case, just to let you know, this is one of the big projects that we're also working on, which is actually mapping the various genomes. We can do this for everything around the world also, same exact way, looking at the genome sequences. And I'm going to just reset this. So that's the sort of mapping that we've been doing for COVID and for looking at the bats and using bioacoustic recordings. And I'd be more than happy to take either questions in the chats. Yes, the sampled Laotian caves are frequented by bat tourists and guano gatherers. There's also one of them that happens to be a sort of religious site. No one's saying that that particular cave, again, you only know what you know from the sampling size. You know, we have very few samples. This is very difficult work to go attempt to capture bats, then to take fecal matter, anal swabs, swipes from saliva, and then go back and analyze them. It's very difficult work that takes a long time. So there's a lot of different programs going on right now that are actually sampling them. But again, the Geographic range of these bats and where they're actually found is very limited. So where the spillover took place is somewhat unknown. But if we do think that the first super spreader event is in Wuhan, it's perfectly reasonable that we can have any one of those tourists or religious folks or someone who came in contact with an infected bat in some way come into the market and be a super spreader event. No one really knows. I think the thing that the Laotian study, which, as I said, just came out a few weeks ago, not really out, it hasn't been peer reviewed yet. It was only just sent around in preprint to some people to look at. The New York Times just did an article about it a few days ago, but it really is probably the most important piece of information that we have on the actual zoonotic transfer. The fact that those particular bats, and again, we've got these same group of Rhinolophus malleana species that seem to harbor this. The question will be now, how are they immune to it, and how widespread are they? The problem really with any of this sort of thing is, and the reason I titled this thing "It Came from a Blue Sky," that comes from Camus' novel *The Plague*, where he basically says, everyone knows that pandemics are possible, but no one really believes in them when they come from a blue sky. This is the sort of thing we just don't know enough about the animals that are zoonotic, are possible zoonotic transfers. We don't know what the next one is. David Quammen's book Spillover is a great book if you want to read about sort of the history of zoonotic transfer, HIV, Ebola. We still don't know. There are theories about particular bats that harbor the Ebola virus, but we still don't know what the zoonotic transfer host is for Ebola. There are ideas, but we still don't know. This is going to take years to sort out precisely, but this study from Laos is probably as close to a smoking gun as we're going to get soon, but it really is important. Yeah, that's the question about what is about the bat immune system that allows them to harbor viruses. That's a very good question. One could ask that about lots and lots of animal viruses that are hosted by animals that don't affect them. We have viruses in us that we host that don't affect us. Bats seem to have an incredibly super immune system when it comes to certain types of viruses, and no one knows why. There are some theories that this came about from the evolution of flight. Now, of course, bats are the only mammals that can fly, and so that and the fact that they are in tight quarters with one another mean that they can fly in and out of other communities, and they come back together. They roost very close to one another. So there may have been an evolutionary pressure in order to develop immunity from a lot of things, just because of their lifestyle and their ecology, but no one really knows. Another question is, who be the source of the virus? Well, it's the source of the first one that we have a genome from, and obviously it's the source of the first breakout. Whether it's the ultimate source is an open question that will probably never actually be answered. But we can at least say that the bats that have the closest genome are in Hubei. They're also in Laos, they're also in India. But we do seem to see that first breakout in the Wuhan region is an important point. So we can't say definitively yes, but it's pretty sure that it is in fact the source of the virus.
John, if you were to sample the bats, let's say in caves today, they would not necessarily be harboring the Delta variant, because that's probably mutating through human to human contact. Is that correct? So would you go back on the tree to where they kind of
exactly?
You go back to the original, the alpha, or
there's a couple. Well, if you look at this is the human tree right here.
Right.
I'm just going to move this chat out of the way. So you can see here that the Delta variant really starts here in September 2020. It breaks off. We've got the epsilon, the beta, the alpha variant cuts all the way back to March of that year. So these are all human sequences. So every one of these dots is a place where a genetic sequence has been done. So we see the location of it, we see the country of the sample, and we see what variant it comes from. So this is from Gambia, actually, this particular sample. So when we look at this, obviously these are all human. The original virus, the native strain, is the thing that obviously jumped over. And then of course, when a virus does make a jump over to humans, it's like the gold standard, because if it is something like a coronavirus, which coronaviruses are acute, they tend to transfer like a lot of RNA viruses through either blood or feces, or in the case here, respiratory particulates. They cause these acute infections, and they want to spread themselves all over the place. But these are definitely human versions. There's something I didn't talk about. There's something called a furin cleavage site, which appears in the human version. Everyone thought this was, something was going to be the smoking gun, that was going to be the thing that sort of nailed down what the animal was. But it appears that furin cleavage site must have evolved in humans also. So the bats don't have it. The virus came into humans and became, mutated in a way that became more susceptible or more transmissible. The thing about RNA viruses is they're RNA viruses. So a DNA is a double helix, an RNA is a single strand. A double helix, when it tears itself apart and recombines, has a way to check itself. In other words, has a way to correct errors. So its mutation rates are slower. RNA viruses, on the other hand, don't have that double check. They don't have that binding back together. They just have the single strand. So to overcome that, they mutate at massive rates. They mutate very quickly. And so that's really part of the problem.
Well, but John,
we can actually look at.
I want to interrupt for a second. If
Go ahead.
you called zoonotic transfer a natural event, but at the same time, it seems like it's the most prescient or most urgent event of our times. So if it was a natural event, why was it so catastrophic? What did we do to cause that natural event, if any? Or is it just part of our society now due to population? Or can you expand on that a little bit? Like the natural event versus the zoonotic transfer.
Well, this was sort of the perfect storm. I mean, RNA viruses, like I said, mutate rapidly. And in the past couple decades, past 50 years, HIV is more than 50 years old. HIV is a zoonotic transfer, Marburg virus, Ebola, Nipah virus. There's a whole slew of them that have happened in the last few decades. This particular one, however, happened to be a coronavirus, which of course is related to the common cold, easily transferred through humans. And they are random events and rare events. Why this one spread so quickly? Well, if you think about the two introductions of the virus, if you think about the way it happened, let me just pop over here for a second. Since we're talking about this, I can. Yes, I don't have that here.
Yeah, when you say natural event, it sounds like it's something that will occur more often, or
it's something that's certainly going to occur. Yes, it's definitely going to occur more.
due to the population, or due to the way the bats have lost their habitat, or what is the cause of
Well, there's a couple things. One, people interacting with animals more, disappearance of habitat where people are interacting with animals in a way that they never have before. Also, the way that we live our lives. So the first introduction and so this is the moment so this is USA WA1, okay? So this is the first moment. That's the first case, the first genome introduction into Washington state. All of the red that you see here are these spread outs into North America, into the West, okay? So this is the first introduction into Washington state. Obviously it happened before this because, what
was
this
the date?
Is January 2020.
Okay,
So this is when the first genome is sequenced. Okay, so obviously the introduction happened before, but this is when the first genome is sequenced. So this is when we know that it's in Washington state. Now, the Washington state one, as everyone knows, is, or at least maybe you don't know, but it came from a visitor from China who came into Washington state. But it's not where the first introduction from New York came from, okay? So if we look, sorry. Back to my sequences. Okay, so if we look at the other side of the phylogenetic tree, so basically this part down here, in other words, on the bottom side here, okay? So this is in January of 2020. We now have a branch that's coming off. This is in January 2020. So this is a branch coming off this larger Wuhan branch. All of this purple is the larger Wuhan branch, okay? We've got this branch coming off, which is a French branch. This is the introduction into France. So these are the European versions that are coming from Wuhan. If we go further, we can see these are the two branch mutations that come into New York. This is the branch originating in France. This is the one coming into New York City. So the ones from New York City were introduced from Europe. So even though at this moment in time, so we're talking about February 2020, the Trump administration had closed down all travel from China, it didn't close down all travel from Europe. So the European version came in with Europeans as they came into New York City. And that's where the New York versions came from. So part of the spread problem is, first of all, the way this virus acts is somewhat different than a lot of viruses. Obviously, we've got this this idea that there's this time period where people can infect others but aren't symptomatic. We have asymptomatic people who can infect others, and so there's this unknown. You know, if you have Ebola, you know, you kind of know it pretty quickly. And so it's just a different thing. And these are obviously increasing. We're going to see more and more of this as habitats change, as people move around more into other habitats. This idea of bushmeat, this idea of those kind of things. Obviously, the whole Wuhan market thing very early on really got this panic going about the various exotic animals that may or may not be consumed and sold in Chinese markets. There was a whole kind of political aspect of it that influenced our reaction to it. And even now, I mean, the political reaction to the virus, to masking, to vaccine mandates, you know, these things are, on the one hand, completely natural events, and on the other hand, this is the really first pandemic of the globalized age. The first pandemic that has a virus that has a high transmission rate, that one does not have to interact with the person directly for it to be transferred, unlike something like HIV, for example. And it's also the first pandemic of the information age. So we know more about this pandemic than ever before. We unfortunately have more false information about any disease than ever before. And so there's a whole mix of things that have made this kind of into the perfect storm. But we really don't know a lot about these animals or these viruses. And, you know, if you read the David Quammen book, and where he goes through the various zoonotic spillovers that have happened in the past, you know, Laurie Garrett, a lot of these people who've kind of written books on, you know, the coming plague was her book from many years ago. You know, people do believe we're going to see more and more of this. Whether it's to this level, probably not. If you look at what happened with SARS COVID-1, it infected a bunch of people, but then it died out fairly quickly. A lot of these viruses will also mutate themselves kind of out of existence. Mutations in RNA viruses are not only always beneficial, but the more human contact there is, the more traveling around during these periods, the more we're going to have this. We're going to be studying this virus. We have so much data. The data that I'm showing here, the being able to weed through millions of genomic sequences, all of which can be traced back to a particular geographic location in a particular moment in time. And then to be able to study how every one of those genome sequences changed down to the, you know, down to the base pair level, really makes this, you know, as I said, we're going to know a lot more about what this means than we ever have for anything else. So
John,
that at least gives.
Just to make sure I understand, did the first crossover only occur after the 5 recombinations you referenced earlier?
That's what people think. Yes, there seem to be 5 recombinations. The Laotian bats seem to point to 5 recombination events that took place between the various species and produced a virus that has 16 of 17 of the amino acid structures that will allow it to bind very efficiently to the ACE2. There may be another one, you know, that's just the genomes from those particular bats. There may have been, you know, another, you know, there may be something closer, you know, obviously, you know, a lot of these bats have these COVID-like viruses, these SARS-like viruses, and they're interacting right now, all of them, you know, millions and millions of them all over the place are interacting. There's recombination of these viruses taking place. And when you think you've got, you know, millions and millions of bats, there's only a small percentage that are actually infected at any given time. But when you've got millions and millions interacting over and over, you've got recombination events taking place, you know, you're just tossing the randomized dice. But a lot of things have to happen. I mean, you've got to have these random recombination events that take place that make the virus, you know, in a certain structure, and then an infected bat has to come in contact with a human being. So there has to be a lot of these sort of not super probable events, but when all of those probable events come together, you've got what we have now. So,
Are you able to tell the transmissibility or the like R naught of the recombining viruses, if you will? In other words,
in the bats, no. You know, there's just not enough of a sample, and also now R values become very difficult because, you know, an R value is basically measured from a population that hasn't seen the virus and now without antibodies. So now there's several different types of R value that people are working with, because now the R value now is obviously very different than it was when no one had any immunity to the virus whatsoever. And so what the real R value is for SARS-CoV-2 is a number of different R values. Those are always based on where you're taking the R value in the time sequence. But as far as the bat transmissibility, no one knows. I mean, when we're talking about even the Laotian bats, I mean, we're talking about, you know, how many millions of these 3 species of bats are there in Laos and India and all of the rest of the area where these bats live. And, you know, we have what, 150 samples. I mean, it's not like, and to a certain extent, the researchers are getting lucky. You know, you don't know what's in the next bat. You know, I mean, you know, if the 2 bats that you know showed these great results weren't in the sample you took, well, you know, then you got to go back but we just don't know. It's you know, we just don't know, and I think that's you know, that's an open question. So. And you know, there's a question about, might these viruses be beneficial? Well, no one knows that either, because we just don't know enough about these bats and their lifestyle and what they do. you know, even the echolocation calls that we're talking about, you know, these are not one-dimensional. The bats will call, children bats will have distress calls when their parents fly away from them. There's social calls where bats that know each other greet each other with a particular type of call. You know, the echolocation calls are obviously different when they're flying towards a prey, so they get much faster as they get closer to an object. And you know, even how these things are really processed in the bat brain. What is the cognitive, you know, what are bats doing? What is their entire lifestyle? Is really it's still very unknown. So they're still very mysterious animals in a number of ways. But I will tell you that at this point there are lots and lots of people who are now studying bats and looking at bat immune systems and trying to figure out what makes them so special and why have this serious group of diseases jumped from bats. Now, of course, HIV is not a bat, one of the bat ones. I mean, HIV is a chimpanzee disease, so that's a little bit different. So anyway, I want to thank everyone. No,
Thank you,
domestic
John.
bats have not factored in the spread. There are, as far as we know, there is the possibility, and people are looking into this. There's the reverse crossover. In other words, that human beings can infect animals with it.
We'd have to fight
Those
back.
studies are being done right now. You know, there's some mink farms in the Netherlands where people think that might have happened, but it's possible that it can go both ways. But again, it's one of those things that really hasn't really been looked into. You know, this was kind of a wake-up call. And the other thing about this is that this was an easy one. You know, this one happened to be a coronavirus. Spike protein hangs out there. You sterically hinder it. We knew a lot about how to do that. We knew a lot about how to make those proteins. You know, there's lots of possibilities of, you know, tons and tons of viruses where we don't know how to make a vaccine quickly. HIV is, you know, a typical one that we could talk about, you know, forever. And mapping the spread of that has been a fantastically interesting and phenomenal thing. That's another talk. Anyway, I want to thank everyone. Like I said, not a lot of maps, but a lot of info.
Thank you so much, John. It was
Great.
so informative. I'm gonna have to read my notes again tonight. Really appreciate you taking the time.
Sure, it was my pleasure, and everybody stay safe.
No, thank you so much, John. And look, we appreciate what you're doing, kind of help dig beneath the whole problem to understand what's happening, where this is coming from, and you know, maybe they will help us to find, you know, either cures for other pandemics, or you know, or more quickly isolate the source, and maybe there's some way to kind of find a vaccine or an antidote at some point with this research.
Yeah, and 100 years from now, maybe somebody will have a bat map on the wall behind them there.
They will, though. We have the beaver map, we have the bat map, you know. Well, look, thanks again, everyone,
that's right,
for joining. If you guys, please check out our website for upcoming events or social media, our Instagram or Facebook page. And if you're not a member, I'm going to encourage you to all join the New York Map Society, so you can enjoy wonderful presentations like this from John, and share your interest in maps with the rest of us. So thank you very much. Have a good evening.
Good night.
Bye.
Everybody.